Good evening, everyone. Thank you for joining us tonight for our RiverWatch Fall Speaker Series. For those of you who are not familiar with the Illinois RiverWatch Network, RiverWatch is a community science organization that trains volunteers to monitor water quality in their own communities. The mission of RiverWatch is to safeguard the future of Illinois rivers and streams through stewardship, education, and sound science. Before we get started, I want to ask that you keep your microphone muted during the presentation to limit any distractions. We will have a Q&A session after the presentation, so if you have questions along the way, you can either put it in the chat box to be answered at the end, or you can save your questions to ask out loud. And with that, I would like to welcome Natalie Hernandez. Natalie is a PhD candidate in the Department of Earth and Atmospheric Sciences at St. Louis University. She has a Bachelor of Science in Environmental Science from Baylor University and has been studying microplastics in the St. Louis region since 2020. Tonight, Natalie will present some of the ongoing research she's conducting as part of her dissertation work with the Hasenmuller Environmental Geochemistry and Hydrology Lab at St. Louis University. And with that, I would like to welcome Natalie.
Thank you. Hopefully you can hear me well, but let me know if there's any audio issues.
Sounds great.
Okay, thank you. Thank you for the introduction. So I will be talking about some research on microplastics in the St. Louis area watersheds. And I want to acknowledge some of the funding sources for this work that are on the screen. And also, I am part of the Hasenmuller Environmental Geochemistry and Hydrology Lab, so I want to thank the members of that lab who have helped me with some of the field sampling and analysis and things that have happened for these projects. So I'll start with some introduction on microplastics just to get everybody on the same page, but then I'll be talking about two different projects. All right, so I'll start with let me get a laser pointer. I'll start with macroplastics. So if you've been out in any local streams, probably wherever you are, but definitely in the St. Louis region, you've probably seen plastic pollution, unfortunately. We use a lot of plastic these days. There's a lot of reasons for that. There's versatility to plastics, durability. They're useful in a lot of different kinds of products, like some of the things that are in the graphic on the right. But unfortunately, this means that there's also a lot of plastic waste that's generated, and this is going into the environment. And one of the benefits of plastics, that's a reason that we use so much plastic, its durability is also a reason that we see a lot of persistence of plastics when they do get in the environment. And we see them in this visible kind of macro scale, but we also see them breaking down into smaller size plastics. And so we see a lot of microplastics accumulating in the environment also. And microplastics, they can be from the breakdown of big plastic items like plastic bottle in this graphic breaking down, but they can also be plastics that are produced in a micro size. So micro beads that are in consumer products sometimes, and then also our clothing is often made of, well, all our clothing and textiles are made of microfibers, and so those fibers can shed from clothes. And that's what I have pictures of at the bottom of the slide, because microfibers are one of the common things that we do see in the St. Louis area. And the reason that we're concerned about microplastics, the two main reasons is that we're concerned about the ecological impacts and also the human health impacts of them entering the environment and becoming more persistent in the environment. And we've seen evidence that there's ingestion of microplastics by a lot of different species. And also they're present in things that we ingest, our food and water. And even more recently, there's more and more studies and news about microplastics found in humans. So that's concerning. And one of the reasons that we're concerned is when we look at studies of organisms and organism exposure to microplastics in lab conditions, we see all kinds of different impacts, a lot of sublethal impacts like growth reduction or reproductive impacts, but also sometimes lethal effects at like high doses. And these impacts that microplastics have on organisms are kind of twofold. There's physical impacts of just ingesting plastic. For example, it can fill the fish's stomach, and then the fish doesn't eat enough food, and then that has kind of physical effect. But there's also the chemicals associated with the polymer itself, any kind of dyes or treatments added to it. So if you think about a clothing fiber, it's probably dyed a particular color. Um, and then also additional chemicals absorbed from the environment. So there's a physical impact and the chemical impact, and it's all different depending on the size of the particle and its shape, and kind of how it's moved through the environment, what chemicals it might have absorbed, etc. So there's a lot of complex things to think about when we're trying to understand the impact of microplastics we're seeing in the environment. So I'm not going to talk a lot about microplastic characteristics, but I will mention them later. And one of the reasons that we're looking at the characteristics is because different characteristics can mean different effects on organisms. So we want to better understand where microplastics are coming from, how they're moving through the environment, their fate, in the hopes that someday maybe regulation or like management efforts will be informed by this kind of research. And urban freshwater systems in particular probably transport higher levels of microplastics. There's been quite a few studies that find this. And we also see in the St. Louis area, especially, evidence of higher amounts of macroplastics and other kinds of litter transported by our urban freshwater systems. There's a lot of reasons for this, just where there's higher populations, there are more people using and disposing of plastic sometimes, and sometimes improperly disposing of them. But there's also things like enhanced urban runoff. So because we have higher amounts of pavement and parking lots and things that water runs off of after rainfall instead of kind of permeating into sediment or soils like it would in a natural area, that means that we have enhanced runoff of any debris that's on the sidewalk or pavement into the creek during flood conditions. So there's kind of enhanced transport mechanisms in addition to sourcing in urban environments. And then we have things like more cars on the road around the city, so more tire wear. And we also have a lot of water that has to be dealt with, municipal wastewater systems, and where there is wastewater treatment plant systems, there's also potential for leaks into the environment, or in St. Louis history, sometimes intentional overflows. And these could contain a lot of microplastics and especially microfibers from our laundry effluent, for example. So there's a lot of reasons that urban areas are important to look at when we're thinking about microplastics. And in the St. Louis area, we're also in a city that is, most of our waterways are feeding, actually all of our waterways are feeding into the Mississippi River. So we're in an important place kind of in the global transport of plastics from cities out to larger freshwater bodies and eventually the ocean. So the projects I'm talking about today are kind of trying to understand the question of how are microplastics distributed across watersheds in the St. Louis region. And I'll be talking about two different ones. The first one is a published paper, so I'll touch on it a little, but you can read more about it also if you're interested. And that is looking at the Merrimack River watershed in sediments. And then the second project I'll talk about is ongoing work that I'm doing for my dissertation research. That's in the Deer Creek watershed, which is a smaller watershed closer to the city. And I'm looking at a variety of different environmental compartments for that work. So first, for the Merrimack paper, the question that we set out to answer for this was, can hydrologic or anthropogenic impact data be used to predict microplastic levels in benthic sediments? So at the time of this project, which was kind of developed in 2017, 2018, a lot of studies were still very focused on just answering the question of where are plastics, like they are in this place, and now we found them in this place, etc. So we kind of wanted to answer that question in the St. Louis area, but also take it a step further and look at some additional data sets and try and see if we can understand a spatial distribution of microplastics that might be related to some of these other data sets. And that's hydrologic data, sediment load and discharge, and then also anthropogenic or human impact data And we looked at land use and point source pollution inputs for that. So as I mentioned, we sampled sediment from 19 sites in the Merrimack River watershed. Those are the black dots in this map here. And you can see also the land use of the watershed. So darker red colors in these land use maps is more urban, and then green is more rural, typically forested or sometimes pasture areas. So you can see the Merrimack watershed has, it's a big watershed and takes up a lot of portion of Missouri, and it has a pretty good span of different types of land use. So it's an interesting site to ask this question. One of the questions we're asking is land use impactful? It's also, in addition to having kind of interesting land use characteristics, it's home to some endangered species. So we wanted to answer our question about microplastic distribution, but there's an additional perspective that answering this question lets us know if we should be concerned from an ecological perspective for some of these endangered species that are present in the watershed. And I've got pictures of them on the slide also. So the sites that we selected, they're here on this map again. You can see they're mostly along the main channel of the Merrimack River. There are 2 sub-watersheds that are in here, but we focused on trying to get more sites that covered Merrimack River. And I also have the table with full list of different characteristics for each site for those variables we're trying to look at and compare to microplastics, but I put the ranges at the top just to make it a little easier to see. So we have land use, urban land use ranging from 4 to about 60%. We have a decent range of point source pollution inputs and discharge. The sediment load didn't vary that much between the different sites that we were looking at. But together, kind of the spread of data in this, we were hoping we would be able to identify what factors from these are most influential for microplastic distribution. And in one sense, we're kind of looking at a couple more sourcing related variables, urban area and point source pollution inputs, and then discharge and sediment load being more transport related variables. If there's more water and sediment transported, does that mean more microplastics? So that was kind of where we were coming from with those variables. And what we saw was pretty low levels of microplastics across the Merrimack River watershed. So everything is under 10 particles, which compared to what I'll be talking about in Deer Creek later, is pretty low. But our highest amount is close to the city. And we also see a little bit of a trend kind of increasing towards the city. And maybe a trend with land use to some extent here. So then when we compare to our different types of hydrologic or human impact data, what we found was that the variables that combined best estimated this distribution were the point source pollution input and urban land cover together. So again, you can read more about this in the paper or ask me questions later if you want. I won't go too much into it, but we tried all of these variables in different combinations, and it was the urban land cover and point source pollution that were most important for microplastic distribution. So in one sense, this might kind of suggest that the sourcing related variables seem more important than transport related variables, but we also only looked at one point in time. So if there's variation over time, this study doesn't really capture that. And that's one of the questions that I want to kind of expand into with the rest of my work. But in general, we're seeing that from this study, urbanization plays a big role in microplastic distribution. So then the next question that I'm asking is, can we better understand microplastic sourcing, storage, and transport by looking at another urban watershed and maybe a more urban watershed? So the rest of what I'll be talking about is my dissertation research towards the question of how microplastics partition between different compartments, meaning atmosphere, water, sediment, and things living in the creek. And how is that partitioning changing across space and time? So just trying to look at a more urban watershed and piece together a better understanding of how microplastics move between these different environmental compartments through space over time. And this research is ongoing, so I'm presenting what I have so far, but I'm still working on these questions. So to characterize this partitioning over space and time, I sampled sites within the Deer Creek watershed. And this watershed I have outlined in the white outline on this map. And then the dashed outline around it is the River des Peres watershed. So you can see a few things. The Deer Creek watershed is very close to St. Louis City. Also, it feeds into the Mississippi River via the River des Peres watershed. So again, this has kind of implications for everything we're looking at in this watershed when we're talking about export. The export is meaning eventually to the Mississippi River. And the circles on this map, which you can see a little better in the inset map here, are 10 different sites that I sampled across the watershed for every month for a year. I sampled water and sediment from all of these sites. I also show here that the sites that have white circles are USGS gauge sites. So I'll talk about discharge data from those gauges later, but the black sites are ones where we didn't have a gauge nearby. And water and sediment, again, are taken from all 10 of these sites every month for the year-long period, which is from 2021 to 2022. But I also took the biota compartment, which was Asian clam or Corbicula fluminea, from a single site, which is this one further downstream with a green X. That's just a site that had a high population of Corbicula fluminea. So those were taken every month for the same year-long sampling period just from the one site. And then in addition to that, just this year, we've started sampling atmospheric deposition. So that only started in spring of 2023. I have kind of limited data to present from that, but that sampling is happening at one site in the Deer Creek watershed and one site in the city to get a little bit of spatial resolution there. Also, as I mentioned, the project is ongoing and the sample analysis is ongoing. So I'm presenting 7 months of the 12 months today. So we'll only see kind of a portion of the trends over time, and then the rest of that is still ongoing research. All right. So to show a little bit about the methods to get at microplastics in each of these compartments. So we have a lot of different sample types, and they all go through different processes to get to our microplastic identification stage. So sediment, we have to dry and do a density separation to separate out the plastics from the rocks, often like very rocky sediment in the Deer Creek watershed. And then the resulting supernatant gets filtered. Our Asian clam, we take out the tissue, digest it, and that gets filtered. Water just gets directly filtered from the creek. And then for atmospheric deposition, we have a setup sitting out that collects what deposits onto it over time. That is rinsed into a bottle, and that's filtered. So there's a lot of different processes happening, but essentially everything gets to a stage where it's filtered. And then once it's on a filter, we look at it under the microscope. So the photos you'll see throughout this presentation are from this step where we're identifying the microplastics, taking a picture, writing down characteristics like size, color, etc. And then eventually the final step is polymer analysis to identify the type of plastic. And that's a step that we haven't done yet, so I don't have that data, but that is part of our eventual plan. And with that data, we'll have potentially a better understanding of sourcing by looking at like the plastic types and using that information to understand sourcing. Also, because of these different sampling methods, the units are different between these, so you'll notice that, and I'll point that out on the next slide. And they're not exactly directly comparable, but our best way to try and understand what's happening is through comparing these different compartments. So here's what we're seeing so far. And this is the averages by compartment. So for the year-long study, it's the 7 months of data averaged at all the sites, all the months into one average. And then I have the range from lowest to highest as well. For atmospheric, it's a different time frame. So it's actually only, I believe, like 6 weeks of data in that one. But on average, we're seeing atmospheric and sediment samples containing the highest amount of microplastics. This is a log scale or an exponential scale on the left. And so we're seeing much lower levels of microplastics in water and Asian clam than what we see in sediment and atmosphere, but also note that there's different units here. So for example, like 73 per meter square per day means every day 73 are falling in a meter square spot if you think about the unit. But for sediment, our 72 on average is just what's in 1 kilogram of sediment. So the atmospheric is actually probably more than what we're kind of representing by putting it on the same scale. But yeah, we're seeing higher levels in atmospheric and sediment samples and lower in water and Asian clam. We're seeing mostly fibers and mostly blue and clear particles. And what we can kind of hypothesize and try to learn from these quantities that we're seeing is that maybe atmospheric deposition is a big source to the watershed since we're seeing high levels in the atmosphere. But we see such low levels in water in particular that kind of suggests to me that maybe the microplastics settle pretty quickly into sediment, and sediment is a kind of a storage place in the watershed, which is something that other studies have seen higher levels in sediment than water. And this might be leading to some accumulation of microplastics over time in sediment in the watershed. And for the Asian clam component, so we do see that they're taking up microplastics from the surrounding environment. So you've got our deposition from the atmosphere, probably also input from other sources. Then things settle into the sediment, but they're also taken up by organisms living in the creek like Asian clam. But the levels were only about 0 to 15 per individual. These are small clams, so 15 is maybe a lot, but doesn't seem like particularly high levels when you think about finding like 73 per kilogram or 72 per kilogram in sediment on average. Something that we did notice also is that there are smaller sizes in clam. So this is the typically length, like longest length of whatever particle. Most of them are fibers. So if you measure along the length of the fiber, this is the percentage of microplastics in each of these size bins for longest length. And you can see the lighter colors here are smaller sizes. You can see in this plot that the sediment has much larger sizes than what we're seeing in the clam, which is kind of interesting. We didn't notice a lot of size difference between other compartments, but it's pretty noticeable here between sediment and what the clam is taking up. And this might be some kind of preferential feeding, but because we see kind of mid-sized to larger size microplastics in most of the other compartments, I think it's actually probably more likely that we're seeing evidence that they might take up a larger size particle, but then be breaking it down inside the clam. And so then we see smaller particle sizes in those. This is also important to think about because smaller sizes can be potentially more harmful to organisms. The smaller you go into like nanoplastic range, things can start to pass through cell barriers easier. And so that's why one of the reasons that sizing is important to think about. And I have some example photos also from water. And then the middle column is some that we saw in the clam and sediment. And these are just a few examples, but you can see kind of what a longer, like more tangled looking fiber is in sediment versus smaller things that we saw in some of our clam samples. All right, so this is what we see when we're just comparing compartments, but what do we see when we're trying to look at the spatial distribution across the watershed? So for that portion of the research question, I have these spatial samples for sediment and water. And here I've averaged all of the months together by site. And you can see that in this plot, I've got water and sediment together in the same pie chart, but it's actually kind of hard to tell because the water sliver is so small compared to what is in sediment. And this is what we were seeing with the compartment comparison a couple slides ago. Our quantities in water are just really low compared to what we're seeing in sediment, and it makes it hard to really talk about trends very much with the water samples. But we can see that there's this very clear trend happening with sediment, where we've got lower amounts in the headwaters and higher amounts towards the outlet of Deer Creek. So when you look at sediment and water combined, which is represented by the size of the circles in this watershed map, you really see like a big increase towards the outlet happening. And what could be causing this spatial distribution? So one thing is that this is headwaters to outlet again, so there could be some amount of erosion to deposition. If microplastics are applied evenly across the whole watershed over time, because of erosion to deposition dynamics, you would probably get more deposited down near the bottom of the watershed. I think that that likely explains part of what we're seeing. But we also wanted to look at some of the variables that we examined with the Merrimack paper. So the percent urban land and point source pollution inputs that were important for that spatial distribution. How does that factor in here in the Deer Creek watershed? But you can see that it's kind of hard to assess spatial trends because the watershed is so urban that it's almost all over 99%. So it doesn't really, can't really assess that trend. And then even for point source pollution inputs, this is current. So this is with all of the historic sewer overflows fixed, and we don't really see much difference. So this may mean that what we're seeing is more from erosion to deposition gradient, but it may also just be because we don't have a big enough range of types of land use to look at. So I wanted to look at this data set together with what we saw in the Merrimack watershed. And taken together, we see more strongly that there may be this relationship happening with percent urban land use, especially, and even that it looks almost like there's some inflection point between what we were looking at in the two watersheds, where we go from kind of lower levels to suddenly like a sort of rapid accumulation happening. But here now it's the case that we're kind of missing what's happening between 60 and 90%. So we still don't exactly have the right spread to be able to look at this trend across like a whole range of urban land use. So it's possible that urban land use is really influential here. But another thing that we haven't looked at yet is the atmospheric element. So we did have atmospheric data for 2 points. We had the city point and the point within the watershed. This is a small data set that we just started collecting this year, but we are seeing so far that there's consistently higher atmospheric deposition at the urban site compared to the Deer Creek site, which is more suburban. And I have some example photos too. So you can see, we actually see pretty similar types of things between the sites, but just larger quantities depositing at the urban site than what we're seeing in Deer Creek. And we only have 2 points to look at, so it's a little bit hard to say, but it's possible that since we're seeing higher levels near the city, there's kind of a gradient of atmospheric deposition impacts from city sourcing that's like decreasing as we're moving outwards away from the city. And if that's the case, it also, I can see here that maybe it's decreasing moving across the watershed because that's sort of in a direction of going less urban. And even if you imagine the Merrimack River watershed, which is underneath this map, a similar thing might be happening there where we still have a lot of atmospheric influence causing the distribution that we're seeing. So most likely it's kind of a combination of both things that I just mentioned, a few things that I mentioned so far. Atmospheric deposition, land use, and even erosion to deposition gradient potentially. And we also haven't looked at the time component yet of the question, only the switching between environmental compartments and distribution across space. So what does our temporal distribution look like? So as I said earlier, I'm only showing 7 months of data. And this is just for sediment that we're looking at, and it's averaged for all of the sites in the watershed for each month. And we see a decrease over time happening, and we have kind of from late fall, early winter to spring here. So this is also corresponding with a sort of an increase in discharge, which I'll show in a second as well. And also when we look at the sites that we have USGS gauge discharge for, that's what this plot on the right here is. For each of those sites that have a gauge nearby, we took the sediment data for that month and the average month's discharge, and we see this trend where there is decreasing microplastics with increasing discharge for the 6 of 10 sites that had a USGS gauge. It's not the strongest trend, but it is significant. And so this is kind of suggesting to me that what might be happening is as we have increasing discharge, increasing flow in the creek, the flood conditions can flush microplastics out of the system. And I have a photo here of an example of what floods look like at Deer Creek. They can pick up a lot of sediment. So you can imagine that the water is turning brown from the amount of sediment carried. Probably any microplastics that are stored in that sediment are also getting picked up and moved around. So we might be seeing this change over time because of this kind of flood-related flushing out of the system that's happening. And here I have the plot of discharge for that sample period. And it's got all the ups and downs of essentially every time it floods is one of these spikes. But you see the general upward trend from November to May, the time over which we have data so far, this increasing flow and the decreasing level of microplastics. And again, this has implications because we're feeding into the Mississippi River for kind of global transport. So for this small urban watershed, if we have a lot of microplastics getting exported out of sediment storage, that means that they're going potentially into downstream water bodies. And eventually, in our case, into the ocean. All right, so I presented a lot of different things, but towards our original question, how are microplastics distributed across watersheds in the St. Louis region? So we're seeing that microplastics partition between the atmosphere, water, sediment, and biota, depending on spatially variable human impacts and also temporally variable hydrologic conditions. And I know that's like a big statement, so I'm just going to break down what we've seen so far again. So when it comes to the partitioning between environmental compartments, which was kind of the first question, we see different levels of microplastics in the different compartments. So atmosphere versus water versus biota versus sediment. And by looking at and kind of thinking about quantities in these different compartments, we can also start to understand a little better sourcing and storage mechanisms for microplastics. For example, that atmospheric deposition may be a big source, and also that sediment storage may be a big place where microplastics are accumulating and kind of holding legacy contamination over time. When we think about the spatial distribution, we see that with increasing development, we are seeing increasing amounts of microplastics, especially in sediment, but also in the atmosphere. So this is happening kind of across the Deer Creek watershed. This is happening if we look at the bigger Merrimack River and Deer Creek data set, and potentially atmospheric deposition related to proximity to the city is even influencing some of our distribution in the sediments. And then the final thing that I talked about, the temporal variation. So far, we're seeing flow-related, or we think flow-related changes in microplastics, especially in sediment storage. So we might be having export out of the system when it's more flood conditions and storage in drier conditions. But we have only done this first 7 months. So the idea here is as we finish the next portion of the months, we might see the trend start to go the other direction and see that storage happening if that is what's driving the temporal trend that we're seeing. So on that note, there's a lot of ongoing work to do still. I have 5 more months to finish the rest of the year of water and sediment samples. And then we also have the polymer analysis that's planned to identify plastic types. In addition to this, I have some more sample sets related to this project and other projects, higher resolution temporal samples, so weekly water, biweekly sediment, and also flood samples for water to try and better understand on shorter timescales the time element of microplastic transport. And I'm happy to talk more about that if people are interested. And the atmospheric deposition sampling, that's something that we only started doing this year. So we really want to capture a lot more atmospheric deposition samples, especially to see, we've noticed a spatial trend that there's maybe higher levels in urban areas, but we want to collect more weeks of samples so we can better understand if there's also a temporal trend to atmospheric deposition. So there's a lot more to do. Again, I'll thank the funding sources on the screen. And sometimes a question I get when I give these kinds of presentations is like, what are we supposed to do? This is depressing. There's microplastics everywhere. So I like to put up a slide at the end that's just some ideas of things. There's actually, if you're in the St. Louis area, there's a watershed cleanup that's coming up towards the end of October. There's also ideas about ways you can avoid using plastics, or if that's not accessible, like other things that you can do to kind of lessen the microplastic impact you have. And I'm happy to talk about that if people want to talk about what we should be doing, but I can take questions now.
Thank you, Natalie. That was very, very good. Great to hear about the work that you've been doing. Let's see. And again, if anybody would like to, you are welcome to take yourself off mute and ask Natalie a question, or if you'd rather, you can post a question in the chat and then I can read it out loud. So if anyone's not watching, they'll know what we're answering here. I guess since I don't see anybody, looks like someone took themselves off mute. You have a question, Laura?
Hi, yeah, I have a couple questions. So it looked like you're planning to sample for at least another 5 more months. And I know that there's always, limited funding and limited time. You want to move on to other things at some point. But what do you have plans to do any longer term study of this area?
Yeah, great question. So the year-long project, the samples were actually from 2021, like November 2021 to October 2022. And I probably should have put the year in the plots, but I was presenting the first 7 months of that data set. So the remaining 5 months is samples that have been collected already. But we're just, it's a slow process to analyze samples for microplastics. So it's the analysis part that's left. The ongoing sampling is just atmospheric, and I don't really have an end date for that. So I'm kind of just planning to keep sampling atmospheric deposition for the foreseeable future until I graduate. But we don't have ongoing kind of like regular monitoring. I think that's something that would be good to have. What I'll say is the amounts in water are pretty low over time so far. We would want to see maybe if there's a dramatic increase. But it actually seems to me that the atmospheric sampling on a weekly basis is actually maybe the important thing to keep up, because we have higher levels in that than anywhere else that we're looking. So that is still ongoing, and I can keep updating you about it. But we don't have anything in place to kind of continue after I'm gone, unfortunately.
Thanks.
Right, and we have a couple of questions from Hannah. I'm going to actually switch the order of these. She was wondering, how do you store samples between when you collect them and when you analyze them?
Good question. It depends on the sample. So for water or atmospheric samples, those are both just water, so they're kind of okay to sit. They don't usually have anything growing in them, at least in Deer Creek. The sediment samples we refrigerate, so there's nothing that like grows in them or like things living in them while we're waiting to process them. And then the clam samples, we froze those if we had to wait until later to analyze them. That being said, at this point, all of my samples are on filters. So they've gone through all the preliminary analysis steps and they're just stored on the filters. And it's just the time-consuming part of looking at each of those under the microscope and finding everything that's the 5-month delay.
Okay. Then her other question is, if one were hypothetically designing a community science microplastic program, Do you have any tips on identifying microplastics? And I will add to that, you know, if this is a volunteer project, thoughts about volunteers sampling or working with sediment versus water samples to do this, since it seems to be more in the sediment than in the water.
Yeah, great questions. Um, I think that so there is more in the sediment than the water, and I think that that's probably true most places that you look. That being said, analyzing sediment samples is a more complicated process than water, and there's not nothing in the water. So I think for something that would be accessible for volunteers, water samples is probably a lot easier. And those you can, if you have some way to do filtration or do sieving and then have a surface that they can look at that like all of it went onto, um, I think that that's the best option. I think sediment would probably be too many steps to set up for volunteers unless they're collecting samples and then you take it to the lab and then you bring it back to them later, which sounds like too many steps. I do think something like it wouldn't seem as connected to the creek, but atmospheric sampling, where you just have a funnel into a bottle, that also seems accessible to me for volunteers potentially if they wanted to have that like in their backyard or something. And then as far as identifying microplastics after you have the sample on a filter or on a sieve, um, I think that what might be easiest is just to kind of identify things that are bright colors. Microscope really helps, but magnifying glass, you might be able to see things as well. And then where it's challenging to figure out is something a microplastic or just like a natural material, oftentimes is if you're looking at like clear or black or brown particles, it can be really hard to decide. So what might be useful to still collect good data, but make it a little easier for volunteers, depending on what the goal is, you could just have them identify things that are obviously like a bright like human made color like blue or red and etc. And then you're eliminating the other kinds of particles from your results, but you also might be more confident that they're identifying everything correctly. Hopefully that helps. And you can email me also if you really want to develop something.
All right, thank you. All right, Teresa had a question. The amount of microplastics in the atmosphere is pretty striking. Did you differentiate between dry deposition and wet precipitation, or are you planning on making that differentiation in the future?
Good question. So right now all of the samples are kind of like combined dry and wet. We do probably have some times of year where it's dry, where there wasn't any rainfall. I don't think we're going to, for the atmospheric project, separate out wet and dry, like do different collections to distinguish those. But what we will do is compare like the amounts we find in our samples when we have a high enough set of samples to look at it over time. We'll compare that to like rainfall and other meteorologic conditions, rainfall, snowfall, different like wind speed and things like that. And so we might see some impacts of the wet versus dry just through looking at that relationship. But we're not specifically collecting only dry or only wet. Exactly.
Okay. And then another question from, well, a series of questions maybe from Laura. You mentioned the time commitment of analyzing samples, and I wondered if you were able to identify fibers to source, just coming from clothing or other sources like fishing line.
No, I think that there's like some times that you might have a suspicion of what you're seeing. Um, some things it's like if I look at my shirt under the microscope and then I look at fibers that I'm seeing in the sample and I can recognize that it looks like a clothing fiber. That does happen, but I think that we're not really going to say for sure where something is from until we do the final step of polymer analysis. And then we'll say what kind of plastic it is, and then we might be able to say what the source might be. Um, what I'd say is like the more you look at the samples, you do start to see things that look like they might be from clothes versus from something else. But that is subjective. So we're just going to use polymer data to officially say that someday.
Okay, and then she also commented, great presentation, really enjoyed it. And then she passes along a link to another potential place to share your research if you might want to copy that out of the out of the chat at some point.
Thank you.
All right. Anyone else have questions or thoughts to share? All right. Well, once again, thank you for a great presentation, and thanks to all of you for joining us tonight. Before you go, I wanted to mention that our next Fall Speaker Series event will be Wednesday, October 25th at 6 p.m. Staff from the Conservation Foundation will be talking with us about how communities are reducing their impacts on water quality by using less road salt. Also, with the approach of the winter season, we do have several chloride monitoring training workshops coming up in October and November. These workshops are 1 hour long virtual presentations that will teach you about the hazards of road salt and how to monitor chloride in our streams as part of our Winter Chloride Watchers program. You can register for this and other future activities on our webpage or from our Facebook page. And that's all we've got here. So thank you again very much and have a great night.